Hi, Tara. Just returning your call.
Lenny, the man, the myth, the legend. So listen, unfortunately, we are gonna need to scrap the car chase sequence from this week's lesson. Yeah, that Star Trek bit from the last episode just completely drained our budget. So sorry, but love ya.
Oh, I'm so disappointed. I really wanted to drive that Ferrari. Oh well. Love ya too. I guess we can just start with a story. A woman is walking along a beach and she sees a talking frog. The frog says to her, "Kiss me and I'll become a drug developer. Marry you and you'll be rich and live the lush life." She bends down, picks up the frog, and puts the frog in her pocket and continues to walk along the beach. A gentleman who is watching her doesn't understand what's happening. He walks over to her and he says, "I heard what the frog said to you, but you put the frog in your pocket and kept walking along. Why?" The frog said, "If you kissed him, he would become a drug developer and you would be wealthy." The woman looked at him and said, "In this brutal biotech environment, I'll make more money with a talking frog than a drug developer." Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. My goal is to explain what vaults are to non-scientists. In my last video, I introduced the strategies that cancer cells use to evade the immune system. I also introduced Steve Dubinet and told you about Steve's attempts to make a tumor attract the immune system using a powerful immune signaling peptide called CCL21. Today, I want to cover three things. First, I'll describe the vault we made to deliver CCL21. Second, I'll show you the results of our experiments to measure the activity of this vault. And finally, I'll describe an interesting approach to determining the mechanism of action of this vault. Stick around, and I'll show you how a glow-in-the-dark fluorescent mouse was invaluable to our studies. After several exploratory discussions with Steve, we decided to start a collaboration to see if we could make vaults that could deliver CCL21 to a tumor, thereby avoiding the problems and expense of engineering patients' cells. You see, your vaults and my vaults are identical, so the CCL21 vaults could be stored and used when needed. You wouldn't need to wait for therapy. They would also be consistent, and the dose of CCL21 could be controlled. All these things would lead to a much reduced treatment cost and a faster response time, as CCL21 vaults would be an off-the-shelf therapy. A postdoc in my lab, Upendra Kaur, worked with Val and Steve's lab, and Shervin Sharma, a longtime collaborator of Steve's and an expert at carrying out mouse tumor model experiments. We used the insect cell protein expression system I described in episode 10 to make a fusion protein joining CCL21 and the INT packaging domain. We then packaged the CCL21 INT fusion protein into vaults, and Shervin tested them in the lung tumor mouse model, which I described in the last episode. We were very excited to see that the vault containing CCL21 INT, we called it the CCL21 vault, was effective at shrinking the tumors. In this experiment, the tumor cells were inoculated under the skin of the mouse, and 9 days later, when the tumors were very small, Shervin inoculated half of the mice with empty vaults and half with CCL21 vaults. You can see in the graph that over the course of the next 10 days, there was a dramatic slowing of the tumor growth in the mice that received the CCL21 vault, while the tumors in the mice receiving the empty vault were not slowed in their growth at all. In fact, Shervin went on to show that one dose of the CCL21 vault was as effective as multiple doses of CCL21 protein and equal to the engineered mouse DCs expressing CCL21 at slowing the growth of this tumor. I told you about these cells in the last episode. When we examined the mechanism of the tumor shrinking, we found out that the CCL21 vault was working by activating the mouse immune system to mount a T-cell attack on the tumor. Steve's lab had worked out a very clever way of looking at the mouse response to immune therapy. They took advantage of a special genetically engineered mouse strain called the GFP transgenic mouse. This mouse expresses a green fluorescent protein in all of its cells, making the cells of the mouse glow green under the correct fluorescent light. Except for this unusual property, the mouse is completely normal. When the 3LL tumor cells were implanted in a GFP mouse, the tumors that grew were not fluorescent. When a slice of the tumor that was not treated or treated with the empty vault is viewed under a fluorescent microscope after 16 days of growth, it looked like this. Here the tumor was stained with a blue stain that binds to DNA and thus lights up the tumor cell nuclei. The faint green stain is just a normal amount of background fluorescence seen in any cell. When tumors were treated with the CCL21 vault, they were much smaller, and we could see a large number of bright green cells now present in the tumor. Steve's lab showed that these were activated immune cells that were coming into the tumor from the GFP mouse. That's why they were bright green. These cells were attracted to the tumor by the CCL21 vault because the empty vaults did not have any effect on the tumor. These bright green cells were further characterized as CTLs, the tumor-infiltrating lymphocytes I mentioned in the last episode. This was precisely what we hoped to see, as it indicated that the CCL21 vault was stimulating the production of immune cells that could attack the tumor. We were also able to show that the mouse treated with the CCL21 vault induced a systemic anti-tumor immune response. What this means is the immune response was widespread and an anti-tumor reaction that involved activation of immune cells throughout the body. Most of these T cells arise in the spleen of the mouse, and these cells could be isolated and shown to kill tumor cells. This graph shows tumor cell killing by T cells isolated from mice whose tumors were not treated, the blue bar, and mice from tumors treated with empty vaults, the light green bar, and finally from mice whose tumors were treated with the CCL21 vaults, the dark green bar. T cells from the mice treated with CCL21 vaults could kill over 60% of the tumor cells. In a future episode, I'll tell you more about my efforts to bring the CCL21 vault forward as a new cancer drug. I'm Lenny Rome, the Vault Guy. In my next episode, I'll follow up with our general efforts to engineer the vault so it could be used to deliver payloads to any cell, and equally as important, our desire to get vaults into the cytoplasm. If you like these videos, let me know by leaving me a comment. And please subscribe and tell your friends.